sunny Seattle. Uh, which is difficult to say coming from LA. But I'm very impressed with this floor of this building. I just love it. And I think every hospital, academic hospital, should be arranged like this. And in fact, I moved to this little institute called the John Wayne Cancer Institute. There's no actual thing here, is there? That's right, that's this one. This little building here, only 30,000 square foot, across from the nice, beautiful hospital, from a big academic medical center, because this is a place to do translational research. And it's been shown over the last 30 years in terms of immuno-oncology development. And I kind of wanted to do the same thing for brain tumors as efficiently, as effectively as possible. But there is a vision to build more there. And there's about 100, 200,000 square foot complex of new research buildings and clinical buildings in the next 5-10 years. Hopefully I'll be around before, at least before the first one goes up. But on the top floor, I'm going to recommend they do the same thing here, but make it twice as big. So on that note, I think what I've learned in the last 20 years of doing clinical and basic research is that it takes a team, it takes a village to really make a difference. And I think we all here want to make a difference in brain tumors and do it as quickly as possible. And I've appreciated the fact that we do need a team effort to do it. And joining Providence has been very enlightening, and we have developed a team around brain tumors, that Charles alluded to, and that we want to work on. But really, it takes everyone. And one of the things that I've always focused on is innovating and doing new things, and in particular, developing drugs that get into the brain for brain cancers. And we want to do clinical trials as fast as possible, and that's really the main motivations we have. And so we have a department that's really focused on translational research in patients, using patient samples, which is a very multidisciplinary team. And we really want to change the way we do things, the standard of care. And like I said, you need to accelerate, and that's what we do in clinical trials. Do it as efficiently, as intelligently as possible. You heard this morning about how some of the issues that we have in neuro-oncology clinical trials, we don't know whether the drug gets in, we don't know what the right biomarker is, et cetera, and can we do things a little bit better and faster? That's what we're really trying to do as part of our vision for the department. And the reality is, and the reason is that we need to do this is because cancer is complex and brain tumors are even more complex. And you saw this slide, I think someone else earlier presented it, about cancer and all the factors involved: the immune system, metabolism, genetics, angiogenesis, and many other factors. And we really need to understand cancer from a multi-pronged perspective and try to attack it in multiple ways to really get the outcome we all want. And when you look at these tumors, you see there's tons of different cell types in there: immune cells, blood vessels, stromal cells, and cancer cells. And even within the cancer, there's subtypes of cells. So understanding this heterogeneity takes a lot of work, and that's why you need a broad approach, big data, genomics, systems biology approaches to really untangle and understand this better. But that's what we can do now more than ever before with all the tools in our war chest that places like the Institute of Systems Biology has developed over the past few decades. And our patients want to help. So we collect tumor tissue, we collect blood, spinal fluid, imaging, clinical data from EPIC. This vast amounts of data needs to be incorporated to make this happen as quickly as possible. And so what are the barriers that we have? Besides understanding the disease and collecting information that we need to, there's several things. One is really this tumor heterogeneity. The next is blood-brain barrier. And then the third is really the immune system and our lack of understanding of it in the clinical context, and how we need to do better so that we can optimize therapies. And actually, fourth is we need to make better drugs from the beginning for this cancer and not rely on old drugs made for other things. And here's what this is what motivates all of us, is to see these patients who respond to drugs And this was a patient of mine actually back in Boston at the Dana-Farber in 2004 or 5 that responded to one of the drugs that was mentioned earlier, which was Gleevec, the first targeted therapy developed for chronic myelogenous leukemia. And we had done the study for Gleevec that we knew did not work. Only about 4% responded. But this patient, for some reason, responded. This is before Avastin. And what you can see is within a month, very robust response. So this is what's called off-label treatments. And that actually started a whole project in my lab. We found the biomarker, which is PDGFR amplification, which is only in a small subset of patients. And we've done a clinical trial at UCSD with the newer generation called nilotinib. And we have a good more than 4% response rate at this stage. And some of the patients are over 2 years out with this drug if you select the right patient in this recurrent glioblastoma trial. And over the years, when you start parsing out each cancer, like was shown earlier with Nitin's talk, that you get into 1 out of 10, 1 out of 20 with a particular biomarker. And so in the clinic, we're faced with this heterogeneity on a day-to-day basis, and we have to make decisions because we don't have a lot of drugs and we don't have a lot of trials for each individual patient. But we do the best we can. And here's an example of a patient who is unmethylated glioblastoma, which is the worst kind of glioblastoma. And you can see the patient was given Temodar radiation and progressed and had another surgery. Patient had profiling next-gen sequencing that showed EGFR mutation. Patient was given Afinitor Tarceva off-label, and patient actually responded for some period of time, shrinkage, and then it eventually grew back after 6 to 8 months, which tells you that resistant mechanisms can come into play very quickly. In other cases that have been published within weeks, you can see emergence of resistance of the cancer. Anyway, we did another surgery, and we found out the mutation disappeared, and another mutation occurred. Regardless, the patient lived overall 34 months, almost 3 years, for an unmethylated glioblastoma, which median survival is less than a year. So this type of information is very important, but we need to do it in a very organized way. And here's another example, interestingly, of a patient. Again, you can see a left temporal glioblastoma that progressed after just 7 months of the standard treatment. And this patient we knew had a BRAF mutation, which is a mutation found in melanoma, and there are drugs available for that. And so we treated that patient with Zelboraf as well as irinotecan, which was another marker that suggested that the patient may respond. And this patient responded and lived for over 2 years out. So there is some hint that this precision medicine approach may have a role on an individual basis. And the difficulty is that everyone's tumors are different types and numbers of mutations. And that's where it's very difficult to study this in a very prospective way easily. But there are people trying to study this with what's called bucket trials, where you profile the tumor and give drugs that you think work for that particular subtype of tumor. And you'll hear more about this over time. What are the other barriers? The other barrier really is to understand that there is a thing called the blood-brain barrier, and drugs don't get in. And the standard doses that are used for lung cancer, breast cancer, are not going to apply to brain tumors, even though the same mutation exists. It's very complicated, and it's very difficult to measure. And Temodar works because it actually does get in 20% through the blood-brain barrier. Most of the drugs don't get in that well. But if you can make a drug get in through the blood-brain barrier better, as in this case, this drug is called ANG 1005, which is Taxol. This is the drug that's used for breast cancer. It doesn't work for brain cancer, but when you conjugate it in this way, the way this company did, and get it across the blood-brain barrier through a special transport mechanism, you can see responses in the brain. And in fact, this is a recurrent glioma patient that we had on clinical trial, and you can see the tumor shrank within a few months of treatment. And this patient, I think, responded for over 6 months with a drug that gets in through the blood-brain barrier. So if you can make a drug get into the brain, that chance of success is much higher. And what about the immune system? Now, this is something of interest for the past decade, and in particular in the past 5 years, and you'll hear more about it in the next session, where it's really amazing how much we're now becoming aware of how important the immune system really is. And in the last 5 years, there's been multiple drugs made for other cancers, melanoma, bladder, lung, and that are showing amazing responses. Patients with metastatic melanoma had less than a 10% 5-year survival before these new drugs are on board, and now they have over 40% 5-year survival with combination of immunotherapies. So it's really changed the field in melanoma and now in lung, bladder, and other cancers. And we're just starting to get evidence that there may be some activity in brain tumors as well. And you heard some of the earlier information about the vaccines and other immunotherapy approaches. But one of the problems that we have to deal with in neuro-oncology is the fact that because brain tumors cause swelling and symptoms, most of these patients are on steroids. Besides the tumor inhibiting the immune system, the steroids that we use also inhibit the immune system and prevent us from effectively using the immunotherapy. As well, the radiation chemotherapy also knock down the immune system. But like I said, over the years, we have used and studied various approaches at immunotherapy, from vaccines to small molecule agonists, and also the checkpoint inhibitors, and more recently, CAR T cells. I think everyone's heard about the City of Hope one patient case report that was published in the New England Journal of using CAR T cells to kill glioblastoma, which had a reasonable response rate initially. The reality is there's nothing approved. We've tried them all, and steroids is one of the limiting problems. And the other problem with immunotherapy in the brain is immunotherapy elicits an immune response, and immune response in the brain means swelling, which means headaches and neurological symptoms. So it's a very difficult balance to manage. Nonetheless, we have seen examples of responses. As I mentioned with the CAR T study, this is a patient of mine with lymphoma, which is a subtype of a brain tumor, CNS lymphoma, primary CNS lymphoma, that occurs only in the brain. And this patient had failed everything under the kitchen sink. I've taken care of him for 8 years now. And when nivolumab was available, we started that as a compassionate use at the time in 2015. And within a month, the patient's tumor responded dramatically, and he's still good a year later now. So you can see, even in the brain, with minimal toxicity, tremendous response if you pick the right cancer type. And when we've tried in glioblastoma and other tumors, and we are seeing some hints of activity, but also a lot of toxicity, and it's not clear. I think we'll get some data maybe at ASCO or SNO about whether there is some efficacy or not with the checkpoint inhibitors. But here's a study that was published a few months, or last year rather, from the Toronto group, where they looked at a specific type of glioblastoma that was a genetically based glioblastoma. And these patients had a genetic defect in a mismatch repair gene that caused a lot of mutations, which means a lot of neoantigens, a lot of foreign antigens that the immune system can see and respond to. So if you look at that on the left, the blue bars, these patients had a variety of glioblastomas, medulloblastomas, and other cancers, and the mutation load is so much higher than patients who didn't have this genetic disorder, even more than melanoma patients that respond to immunotherapy. So when they treated these patients with the checkpoint inhibitors, and as shown in the slide here, you can see that initially, before treatment and then after treatment, you can see inflammation, swelling, and then shrink or improvement in swelling over time, as shown here. And the contrast enhancements as well. More enhancement and then less enhancement. So this highlights that in certain cases, even in glioblastoma, there is a possibility of response. There is a possibility of using the immune system in certain patients that have the right genotype. The only issue is you need to be able to control the inflammation other ways, whether it's Avastin or steroid-sparing type of approaches. This is a slide from a colleague, Hideo Okada, who's an immunologist at UCSF now. And even in grade 2 tumors, which we don't think of as being very immunogenic, you can actually see immune cell trafficking in the brain, which is always a question. You know, brain is immune privileged, and can you actually get an immune response in the brain, especially when the blood-brain barrier is intact? And here you can see a grade 2 tumor shown on the flare, doesn't enhance. And after vaccine with his approach, you can see more swelling. Patient is asymptomatic. You can also see enhancement during this time, a few weeks after treatment, and then resolution of the enhancement. And this means that, to me, we don't know what the efficacy, long-term efficacy is yet, but this means to me that even in a grade 2 non-enhancing tumor, that vaccines and other immunotherapy approaches can see the brain, can target the brain, and at least traffic enough to be able to do something in the brain without toxicity. So it's really a balancing act here. And we're interested in figuring out which patients, and this is something completely new. We talked a lot about precision medicine, about identifying genetic phenotype and finding the right drug for the right patient. But for the immune system, we need to have those markers as well. We're talking about PD-1, PD-L1, but there are many other markers in the immune system that we need to think about and figure out. And I suspect in the next few years, you'll see bucket trials for immunotherapy that are based on markers as we understand this process a little bit better. And that's actually one of our goals for the SPORE project, is to figure that out. So again, we go back to, it's really important to collect information from every patient. We need to be able to get tissue, we need to be able to get blood, CSF, et cetera, and really do this in a very organized way in order to make progress faster. And we certainly, as what Charles showed, we have many trials related to drugs, related to immunotherapy approaches, and targeted therapy approaches that we have ongoing. I'll just skip these for right now. So what I thought I would do with the rest of the time is really talk about, I told you 3 main things, which is the blood-brain barrier, the immune system, and precision medicine. The fourth is really what it goes back to my lab, is really developing new drugs. One of the things that we've understood is that there are stem cells in these tumors, the resistant, slow-growing resistant cells that after treatment with our standard therapies, these cells still persist. And you've heard some talks about those cells and what are the molecular drivers. But how can we target this better? And that was actually the basis for my postdoc at Dana-Farber, working with Chuck Stiles. He had discovered a family of genes called the olig-1 and 2 genes that are important for brain development. These are transcription factors, like I said, important for brain development, but they're not active in the adult brain and not usually found outside of normal tissues. But in a variety of studies that I've summarized here that we did over many years, we know that these genes are critical and drive glioblastoma tumorigenesis. They also promote resistance to radiation, and they drive invasion, driving through the brain healthy tissue. And we've also shown that these cancer stem cells in glioblastomas, drive the tumor growth, as well as are, based on a variety of other studies, are immunosuppressive. And as I mentioned, these transcription factors are highly expressed in grade 2 and 3 as well. So the idea is if we can drug that molecule, which I can't, that's a 2-hour talk, which I can't talk about. But if we can get a drug that targets stem cells and combine it with the standard treatment, the goal is to fully eliminate these stem cells and prevent the recurrence of tumor. And I'll show you some examples of our drug development efforts. But if you knock out OLIG2, this is the data showing that when you knock down OLIG2, the cells get wiped out with radiation, even at 2 gray radiation. And And various studies have shown that when you have OLIC2, it does drive tumor invasion. And there's also, like as I mentioned, this molecule is also involved in immunosuppressive phenotype. Based on various studies, we can see with the use of this drug, you can get rid of those IL-17 producing cells when we give the drug to glioblastoma cells. So what, again, summarizing a lot of chemistry that my colleagues at UCSD did over 4 or 5 years, we made drugs that target this transcription factor, that are orally bioavailable, that cross the blood-brain barrier, the brain-to-plasma ratio is above way above 1, and without significant side effects, and these are orally bioavailable. And when we study these drugs in the context of animal models, in immunocompetent GL261, the drug itself has, radiation has some activity, shrinking the tumor here, but the drug has even more activity. And when we combine it with radiation, it has a lot of activity in suppressing the tumor growth. And we can monitor the biomarker OLIC2 levels in the tumor tissue pre-treatment and after treatment, and we can see a dose-dependent effect. So it's really targeting what we think it's targeting, and it has a biological effect. And in fact, it turns out this compound is so good that, that we're doing the IND-enabling studies this year, and we think we can go to the clinic next year. And here's data showing that it also delays tumor growth in orthotopic tumor models. And as I mentioned, we're planning for a phase 1 study next in 2018. And so, as I mentioned, you need a team to make a difference. And what I've appreciated by joining Providence is to join Swedish and Institute of Systems Biology to put our heads together to make a difference. And I think in this era of big data, our goal is to really figure out the complexity of brain cancer from all the different approaches you hear about, genomics, immunology, to proteomics that you heard about from Dr. Hood, and really push that forward into the clinic as quickly as possible. And so between 4 institutions in the Providence system, we have this vision for putting a sport together that we hope will change the way we stratify patients for clinical trials and the treatments that we give patients. And these are just all the people that have funded the research over the years. And I'll close by saying it did rain for 6 weeks in Los Angeles. It may still be raining. But the good thing was that in my backyard, it's hard to see, but there's a full rainbow that I saw. So anyway, there is something at the end of all this rain. So thank you.